Thank you, Anna. Indeed, today I would like to present you novel 7 Tesla MRI markers of small vessel disease and how we've currently characterized these in CADASIL. So when we think of small vessel disease on MRI, we probably think of something like this: lacunar infarcts, white matter hyperintensities, but also microbleeds. And over the years, these lesions have almost become the equivalent of small vessel disease. However, it's important to realize that these lesions are the consequence of small vessel disease. They are the consequence of underlying problems in the small vessels. And we believe that in order to try to understand small vessel disease better, we should really try to study small vessel disease at the level where it occurs. Obviously, it has been very difficult to do so in vivo, given the small size of small vessels and the limitations that we have with MRI. But my aim for this talk is to show you how we are currently trying to characterize biomarkers of small vessel disease at the level of the small vessels themselves with the use of 7 Tesla MRI. 7 Tesla MRI opens up some new possibilities. For example, what we can do is we can zoom in and image individual perforating arteries, for example, in the center semioval and basal ganglia, as you can see here. All the green little circles are individual perforating arteries, and within these arteries, we can measure blood flow velocity and blood flow pulsatility. Another thing we can do is we can look at small vessel reactivity. I think most of us are familiar with the paradigm where a participant is laying in the scanner, looking at a flickering dartboard which activates the occipital lobe. This causes an increase in blood flow to that activated brain area, which we can measure with the BOLD response. You see a typical BOLD response here down below in blue. So the paradigm in itself is nothing new. However, with the use of 7 Tesla MRI, we can really look at the reactivity at the level of the small vessels rather than the larger draining veins. So these two measures are really measures of small vessel function that we can perform in vivo. So what did we do? These two measures, we tested them in a CADASIL cohort. We saw 23 CADASIL patients and 13 control participants. And these were recruited in LMU in Munich in the group of Professor Dichkans. They underwent a baseline day there and then traveled to Utrecht to undergo 7 Tesla MRI. And these are some of their characteristics. But for the sake of time, I'd only like to point out that age and sex were mostly similar between the groups. And if we look at disease severity of our CADASIL cohorts, then we see that all of them had T2 white matter abnormalities, but not all of them had ischemic stroke or mood disturbances or cognitive problems. So they position somewhere in that blue square. Now the interesting part, the results. So first let's have a look at the results of the perforating arteries in the center semioval. These are unpublished data. First of all, when we look at the perforator density, we see that this is highly similar between CADASIL patients and controls. Blood flow velocity, however, is significantly lower, and blood flow pulsatility is significantly higher in CADASIL patients. And you can see that the effect sizes are quite large. Interestingly, in the basal ganglia, we see the exact same pattern. So again, perforator density is similar, blood flow velocity is decreased, and blood flow pulsatility is increased, which we think is a measure of vascular stiffness in this group. Again, effect sizes are really quite large. So we're very excited about these results because we find the exact same thing in 2 regions, and we are therefore really confident that these are robust measures. Of course, there was this other one, small vessel reactivity, that we could measure. I told you that we could make a fit of the average BOLD response in reaction to the visual stimulus. And you see the fits of those here. In blue, the controls, and in red, the patients. And it's not so difficult to see that the amplitude is quite a bit lower in our CADASIL patients, meaning that the increase in blood flow to the activated brain area is significantly lower. Also, the timing of the response seemed to be slightly delayed. Again, the effect sizes are large. So with these 2 measures, we can show that we can, for the first time in vivo, show a functional problem that's occurring at the level of the small vessels themselves. So to already quickly wrap up, I wanted to show you that we think it's important to try to study small vessel disease at the level of the small vessels. And I've tried to show you that there are exciting ways to try to do so when we use 7 Tesla MRI. First of all, I showed you that we can measure blood flow velocity and pulsatility in individual perforating arteries. And also I showed you that we can measure small vessel reactivity. These aren't only interesting disease mechanisms. These are also potentially interesting biomarkers in etiological or treatment studies. So currently what we are doing is we are not only looking at these two markers, but also a third one. We're also assessing reactivity where we use a CO2 stimulus instead of the visual stimulus, which has the asset that we can then study vascular reactivity throughout the entire brain. And we're doing this, as I showed you, in CADASIL patients, but also in patients with sporadic small vessel disease, and also in a population-based cohort in collaboration with the Rotterdam study, really to capture a fuller range of small vessel disease. Many others contributed to the work I presented today, and I'd especially like to acknowledge Tina and Anna, with whom I've performed most of the work that I showed you. These are our funders, and I'm happy to take any questions.